So, hi everyone. Oh, sorry. Get the slides.
Sorry, 1 second, we're having technical difficulties.
Okay. Hi everyone. We're part of the dissonance group under the John Nikia Neuro Lab, and today we'll be presenting on COVID-19 and cognitive dissonance. I'm Tanisha.
My name is Amulia.
And my name is Mano.
All right, so our plan for today is not as straightforward as Colloquium usually is. We're actually going to give you all a chance to participate in our study live tonight. Then we'll take some time to analyze your data and come back at the end of Colloquium and present the data that we collected from you all. Now, Mano's going to explain how you can participate.
So here's how you can participate in our study. All parents of ASDRP researchers have received an email with a consent form to sign their children up for a study, as previously mentioned. This email also contains other information that you may want to know before signing up for a study. About 50 parents have already responded, meaning around 50 students in the Zoom should have already received an email with a link to another consent form, which allows you to participate. If you would like, please check your email now and begin filling out the questionnaire. If you haven't received this email, please check with your parents and make sure that they have filled out the parental consent form. This is extremely important, and we highly stress this. Only researchers with parental consent may participate in this study. Please do not distribute the consent form link that has been sent to you. Attempting to participate without legitimate parental consent will be seen as an attempt to jeopardize the integrity of this research project and violate the code of conduct for our research. If you wish for your data to be a part of tonight's analysis, please fill out the form in the next 30 minutes or so. We will be sending time limit updates in the chat for every 10 minutes, and we'll also be answering any technical difficulty questions. Please be aware that even if you don't want to participate tonight, you can still respond and check through your emails for the questionnaire and consent form. We really appreciate everyone's support and time to participate in our study. However, if you're not comfortable to do so, feel free to ignore our messages. Now let's move on to an introduction of the main subject for tonight's presentation, cognitive dissonance. So what is the cognitive dissonance theory? Cognitive dissonance theory is a theory stating that when one's attitude conflicts with their behavior, a discomfort arises in the brain. We call this discomfort cognitive dissonance. Although attitude and behavior may seem interconnected and similar terms, they're actually two separate concepts in the world of social psychology. Attitude is defined as a person's internal morals or values. They are preconceived beliefs people use to judge their actions and those within the world around them. Behavior is defined as a response to stimuli. Behavior is often directed or guided by attitude, but not always. In a way, you could think of behavior as external and attitude as internal. When there is a discrepancy between behavior and attitude, in other words, between someone's external actions and their internal values don't match, the discrepancy will cause cognitive dissonance. For example, a well-known demonstration of cognitive dissonance theory is known as the carnivore's dilemma. Many people enjoy eating meat or other animal byproducts. However, there's a lot of animal cruelty involved in farming. When people learn of this cruelty, they're faced with cognitive dissonance. Eating meat is good, but the means to obtain the meat is bad. Most people would agree that they're against the suffering of animals. So knowing that they're contributing to that suffering by eating meat causes cognitive dissonance. The attitude that animal cruelty is bad is contradicted by the behavior of eating meat, therefore creating cognitive dissonance between the attitude and the behavior. And this is one of the many everyday examples that we've seen. Cognitive dissonance has also been observed in both humans and animals. In one study, cognitive dissonance was observed in monkeys as well as young children around the age of 4. These results suggested that cognitive dissonance is not societally taught and is instead an ingrained aspect of the brain. Cognitive dissonance theory has received a number of important revisions since its conception. A protege of Festinger, Elliot Aronson, provided an important revision to cognitive dissonance theory in 1960. Aronson expanded on the concept of self-justification and why it is so hard for people to admit their wrongs or incorrect beliefs. He proposed that one of the strongest beliefs someone holds is that they are sensible. The reason that cult members will try to convert others, even after their leaders' predictions have been falsified or their belief system questioned, is due to the person's inability to admit that their beliefs or attitude is wrong. In the 1970s, scientists moved away from cognitive dissonance theory because of the ethical concerns. Specifically, concerns grew over deception and emotional harms as a result of testing cognitive dissonance. In the 1980s, cognitive dissonance began receiving more attention again, as it was used for the basis of many mini-theories, including self-verification and self-affirmation theory, which both relate to the threats on the sense of self. Self-verification refers to a person's desire to be known to others by their good morals, and self-affirmation refers to a person's own desire to see they themselves as morally good. The causes and effects of the perception of these threats were studied in relation to cognitive dissonance. Scientists Festinger and Carl Smith first demonstrated cognitive dissonance in their experiment in 1959. 71 participants were made to do a very boring task for 1 hour. After this, all participants were told to convince another person in the waiting room to engage in the dull task. However, half of the participants were received $20 to do so, and the other half received just $1. So as you can see in the experiment design diagram, the first red box says, "I am not cheap and don't lie easily," which was the internal attitude of the most of the participants. Then they had to lie about their opinion of the boring task, incentivized by either $1 or $20. Those who were paid just $1 had higher dissonance than those who were paid $20. This is because lying for just $1 did in fact make the participants feel cheap, causing high dissonance with their initial attitude. Therefore, they felt a higher change in perception of the task as it was changed from boring to fun. This was because they didn't want to believe that they were cheap and were lying about a boring task. Thus, Festinger and Carl Smith first proved that cognitive dissonance existed through this experiment. Now let's move on to our team study.
So now I'm going to give you all a quick introduction to our study. So here's a little bit on the questionnaire you guys will be taking. It was created on the HIPAA compliant service jot form. The contents of the questionnaire consist of both questions asking for demographic information and the actual survey itself. So essentially within the survey, participants are asked to rate their agreement with the statement, which may be related to topics like mask mandates, vaccination requirements, social distancing at school, and various others on a scale of 1 to 10. 1 meaning that they strongly disagree with the statement, and 10 meaning that they strongly agree with the statement.
So now we'll take a quick break in our presentation to allow more students to fill out the questionnaire. Again, we highly stress that only researchers who have parental consent may participate in the study. So please feel free to take this time to go quickly ask your parents to fill out the consent form if you wish to participate, or else ignore, and we'll be right back as soon as we live analyze your data.
Yoo-hoo! It feels so exciting. Okay, so we go off and they'll work on your data and the other two teams will present. We'll be back.
Thanks, Sahar. I think I have one presenter for tonight as well as Omar. So Kara, are you ready?
Sorry, I was just sorry to interrupt. I was just wondering if the Omar group can go first because we have some people in another time zone and it's getting a bit late. Sorry about that.
I guess rock, paper, scissors.
It's okay. I can go after.
Hey, Edward.
Hey, Omar, how's it going?
Good, how are you?
Sure. Yeah. Why don't you guys go first real quick, and then we'll present.
All right. All right. Go ahead, guys.
So hello everyone, my name is Wesley and I'm from Dr. Amer's group, and we're here to present our research on supplementary cementitious materials. And I'm presenting today with both Max and Angel.
Next slide, please. Oh, hi, I'm Angel.
And I'm Max. Hello.
Cool. So I guess I'll briefly go over what we're presenting today. So first, we'll give a bit of background information on cement industry and how that relates to carbon emissions. And then Max will talk about carbon capture methods and introduce supplementary cementitious materials. And then we'll go into specific types of supplementary cementitious materials and then future research. So next slide, and I'll pass it on to Angel.
Thank you. So I'll begin with an introduction on the cement industry. About 4.1 billion metric tons of cement are produced globally every year, and this contributes to about 8% of global carbon emissions annually. Altogether, those carbon emissions is causing the world to approach 1.5 Celsius increase of global temperature since pre-industrial levels, which is a dangerous threshold. Next slide, please. So a brief background on global warming. So this is caused by the greenhouse effect, and which works by the sun first emitting radiance and heat at the earth, which some of it is reflected, but then those greenhouse gases, which mainly composed of carbon dioxide, creates this greenhouse effect, which means some of those radiance can be reflected back in space, and that warms the earth up. And the Paris Climate Agreement aimed to limit global temperature increase to 1.5 degrees Celsius, but we are already at that temperature increase. Next slide, please. So in the production of concrete and its life cycle in general, a lot of carbon emissions are created. And so cement is the blinder, which is like glue, and aggregate is the filler, which consists of crushed stone, sand, etc. And first, those raw materials such as limestone, silica, alumina, and gypsum needs to be mined. That mining process releases a lot of carbon dioxide, and then it needs to be heated up and to about 1,400 to 2,000 degrees Celsius, and that manufacturing process also releases a lot of carbon dioxide. And then the mixing process and the transportation releases a lot of carbon dioxide as well. And then the use of concrete can absorb a little bit of carbon dioxide, and also the old concrete can absorb it a bit through carbonation, but that process takes decades and a very long time. Next slide, please. So the main contributing factor of carbon emissions from the cement industry is this clicker production and calcination process. It contributes by two ways. First, the kilns needs to be heated up to 1,400 degrees Celsius, and that uses a lot of fossil fuels, which releases carbon dioxide, and second, calcination reaction to create clinkers, which is the intermediary product to produce cement, also creates a lot of carbon dioxide, and the clinkers is used when grounded with additives such as gypsum, which decreases the amount of concrete, and those additives also control setting time and give strength to cement. And the calcination process thermally decomposes calcium carbonate, which is found in limestones, to calcium oxide and carbon dioxide, which is released as a byproduct. Next, Max will talk about the carbon capture process.
Yeah, so thank you for the introduction, Angel. So yes, I'll be talking a bit about the carbon capture methods that can be used in the cement industry. But first of all, carbon capture is defined as pretty much any process where carbon dioxide that would be emitted into the atmosphere is instead captured or stored, so it is not emitted into the atmosphere, not contributing to the greenhouse effect. So one of these carbon capture processes that are commonly used in the cement industry is amine scrubbing, which is using a solution to absorb carbon dioxide after combustion processes. But the downside to this is that it takes a lot of energy, costs a lot, and the carbon dioxide collecting equipment corrodes over time with use. Another method is oxy-fuel combustion, which is instead of combusting fossil fuels in normal air, they instead combust fossil fuels in an atmosphere of pure oxygen, making carbon dioxide easier to capture. But of course, the downside to this is that you will need an additional oxygen supply, and this takes a lot of energy and requires big changes to industrial buildings and power plants that we currently have. We also have commonly used calcium looping, which is using calcium oxide here to capture CO2 and react with it to create calcium carbonate, which is of course good because calcium carbonate is a key ingredient in making cement. And this creates a sort of loop that theoretically can go on forever, but the issue is that as you go through the loop multiple times, the calcium oxide degrades in reactivity, and so unfortunately it is not infinite. And so the method we are focusing on to capture carbon is, of course, the use of supplementary cementitious materials. So we are replacing cement to lower emissions. So SCMs are, quote, an organic material that contributes to the properties of cementitious mixtures through hydraulic or pozzolanic activity, or both. So hydraulic processes are involve the setting and hardening of concrete with water involved in the reaction. And then pozzolanic materials have the ability to react with calcium hydroxide when water is present. And it just so happens that pozzolanic materials are commonly found in industry byproducts such as fly ash, iron slag, and silica fume, which my group mate Wesley will be talking about more specifically later. So how do SCMs work specifically? So when we hydrate cement, what we want to make for the concrete is CSH, or calcium silicate hydrate, highlighted in orange there. But as a byproduct, we also get calcium hydroxide, or CH, highlighted in the red there. And we want CSH because it is the stronger structure, making stronger concrete. But when we add SCMs, they can react with the CH, the weaker CH, to then create more of the stronger CSH that we want. And thus, we create more efficient concrete where we need less cement for the same amount of CSH. And therefore, less cement means less emissions. So here is a brief summary on the ways that SCMs can impact positively carbon emissions. So first of all, of course, as I was saying, when SCMs partially replace cement, of course, you use less cement, and therefore you have less carbon emissions. But also, as I mentioned, SCMs are commonly waste materials from other industrial processes. So reusing and reducing these materials also creates less emissions. But also, lastly, SCMs can improve the properties of cement, such as in the areas of strength or durability. And of course, stronger concrete lasts longer, so you need less cement over time, and so you have less emissions over time. And so, as you see here in these graphs, specific SCMs have varying effects on concrete, some positive, some negative. So we cannot just replace all of cement with SCMs because we have to balance industry standards with the emission reduction properties of SCMs. For example, here you see fly ash class F, which has beneficial properties, but it also increases the setting time of our resulting concrete. So say you're building a building and want to finish within a certain time frame, then you can't only use fly ash class F because that will make the construction process take too long. And so next, my group mate Wesley will be talking more about the specific SCMs that I mentioned earlier.
Yeah, so thanks, Max, for the intro on carbon capture methods and SCM. So the first type of supplementary cementitious material that I would like to talk about is called fly ash. And you see next to it, I have in parentheses ASTM C618. And so essentially ASTM stands for American Society for Testing Materials. And it's essentially an international organization that makes a lot of standards for industry materials, and specifically for fly ash, it's ASTM C618. And so essentially what fly ash is that it's a fine powder-like substance produced as a byproduct of coal combustion. And these can typically replace Portland cement, which is the most common type of cement used around the world. But they can typically replace Portland cement by up to 30 weight%. And it's further divided into two different classes. So class C fly ash, which is higher in calcium concentration and has cementitious properties, can usually replace Portland cement by a range of 20 to 35 weight%. And for class F, it's more of a pozzolan that consists mainly of alumina and silica and can replace Portland cement by a range of 15 to 25 weight%. And the picture on the right is a picture of fly ash. But yeah, next slide. So next SCM that I would like to introduce is called slag, and it's essentially a byproduct of smelting metals and ores like iron and slag, like iron and steel. So it's essentially slag is like a waste material from the iron and steel industry, and you can actually use it as an SCM to replace Portland cement. And research has shown that you can typically replace Portland cement by up to 20 to 50 weight%. And because of that, it has high potential to reduce the carbon footprint of the cement industry. So specific research has shown that using 1 kilogram of slag could reduce carbon emissions by 0.27 kilograms to 0.43 kilograms. Next slide. And so another type of SCM is called silica fume, and it's essentially a waste material from the semiconductor industry. Specifically, it's a byproduct of reducing quartz in electric arc furnaces, and it's a highly pozzolanic material. These typically replace Portland cement by a range of 5 to 10 weight%. and it's usually added to decrease cement concrete permeability and also increase its corrosion resistance. Next slide. And so another type of SCM is called ground glass fibers. And this is, again, a waste material from the glass industry this time. And so essentially, within the glass industry, there's a lot of high standards. And those that don't meet those standards that the customers want are essentially like off spec, so off specifications. And these would generally be disposed of in landfills. But instead, you can actually use them as an SCM. And so these can typically replace Portland cements by up to 20 weight percent. And it also, on top of that, improves durability and resistance against alkali silica reaction. And so essentially what alkali silica reaction is, or ASR in short, essentially the alkali from cement can actually react with the silica from aggregate, which is what you use to create concrete. And so within the pores of the concrete, it creates something called like ASR or alkali silica reaction gel, and it swells up and causes the concrete to expand and eventually crack. So obviously this degrades the concrete over time and is bad. And so what ground glass fibers does is that it inhibits this reaction. So therefore it improves its durability and its sort of lifespan. Next slide. And so lastly, I'd like to introduce flash graphene, which is a relatively less researched type of SCM. And it's produced specifically through a process called flash Joule heating, which essentially is where you take any solid carbonaceous material such as plastic, rubber, or coffee beans, and you subject them to an electrical current, which heats up the carbon material to 3000 degrees Kelvin in about 10 milliseconds. And so essentially all the carbon-carbon atom bonds break and they reform to create graphene, or it's called specifically flash graphene. But research has shown that adding 0.035 weight percent of flash graphene to Portland cements can increase its compressive strength by about 30%. And this is because it encourages CSH formation, which is what Max talks about in one of his previous slides. But yeah, next slide. So currently we've basically completed a literature review and we've been selecting materials that we want to test on. So for example, like fly ash, slag, or ground glass fibers. And sort of our criteria throughout this selection process is one, whether its ability to reduce carbon emissions in the cement industry. So that's one of the factors that we look into, but also if it does not, we would also test them as fibers, which are essentially materials that can improve the mechanical properties of cement concrete. But going back to the high carbon uptake, so assuming that they do reduce carbon emissions, we would also test them as an SCM, their pozzolanic activity. And assuming it has high pozzolanic activity, we would also test their mechanical properties, so the effects that the SCM has on concrete. And then lastly, there's geopolymer concrete, which is essentially a full replacement of cement, so like an 100% weight replacement. And so this would be the ideal situation because you would replace all of the cement, which is where the majority of carbon emissions from the cement industry comes from. So we would test if it has any potential in fully replacing cement, which is sort of our final and last step. But yeah, next slide. So for acknowledgements, we'd like to thank ASDRP and our mentor, Dr. Amer, for guiding us, and for presenters and people who worked on the presentation. So Max, Angel, me, Dia, and Chinmay. And also I would like to shout out our new members, which include Angel, Daryl, Dia, Max, Rashida, and Rishabh. Next slide, which should be the last slide. So thanks everyone for listening. Comments and questions in chat if you guys want to. And then I think a few slides after this are all the references if any of you guys are interested in it. But yeah, that basically concludes the presentation. So to answer a question in chat, or if Edward wanted to.
I was just going to say you have a question in chat.
Okay, yeah, cool. So is it possible to use multiple SCMs in one mixture? And the answer is yes, it's possible to mix them, but it influences a lot of the properties of the cement that you produce. And so you would also have to factor that in. But yeah.
Very great presentation. I love it. It was very structured, very organized. There was a flow in it. I'm very excited to see what you guys are doing. It's very exciting. Good job. Well done. It was very clear what is from starting to end, what is the agenda, what you're going to do. Very good job. Well done. It was a beautiful research project. Nice.
And Omar, I love these cement trucks all over the presentation.
Yeah, thank you so much. Yeah, the guys did a great job.
All right, so Kara is up next. Let's see, Kara, are you there?
Yeah, I'm here.
Great. So our next speaker of the night is Kara Tran, who is a rising senior at Dublin High School. I think when Kara first joined ASDRP when she was a freshman, hopefully you've learned a few things along the way. I've certainly gotten a chance to see Kara grow not only as a scientist, but also as a leader in spearheading now a lot of really cool chemical biology that we're doing in conjunction with the organic synthesis efforts in this group. So without further ado, Kara has a lot of fresh data to share with us. You ready to go, Kara?
Yeah. Okay. So hi, once again, my name is Kara. I'm a rising senior at Dublin High School, and I will be presenting on my work on the chemical biology side of things, specifically mechanistic insights into in vitro models, in this case being cancer cells, for anti-cancer small molecule drug discovery. So just as a brief outline, I'm first going to discuss an introduction into small molecule cancer therapeutics. Then I'll go into some of my work on screening common reducing agents and media that we hypothesized were interfering with the MTT assay, which is a colorimetric cytotoxicity assay commonly used to determine cellular activity and viability in response to a chemical compound. After that, I'll briefly explain some of the chemistry behind our work on minastrol and its analogs, specifically trifluorominastrol, and how we use the MTT assay to get biological activity data determining the cytotoxicity of each compound. Finally, I'll introduce some of our work on antigrapholide, as well as the analogs that we've made of it, and our MTT data on the cytotoxicity of those compounds, as well as introduce the background for our current work to procure RT-qPCR data of genes involved in targeted pathways antigrapholide is known to derive some of its abilities from. So before getting into the specifics, it's important to lay the foundation that drug discovery begins with small molecules. In order to reach this stage of clinical trials, a compound must go through the pathway of creation or extraction to in vitro studies to clinical trials and then onward, which of course starts first with its chemical development and extraction. However, these compounds differ slightly when comparing natural products to synthetic small molecules. Natural products are isolated from nature and not created. Because of this, there is a rich library of existing complex chemical entities within this category ready for screening. However, isolation scalability for mass use as a pharmaceutical might prove to be challenging. Synthetic small molecules are created in a medicinal laboratory and therefore offer opportunities for de novo or from scratch molecular design. However, they come with increased risk of cytotoxicity. Within these categories comes a third approach at drug discovery, which in this Newman and Craig pie chart is labeled as mimics of natural products. This is the realm of semi-synthesis and analog work, where natural products can be altered to their chemical structure with the hope of increasing their biological activity. This takes us to the next stage of drug discovery and the road to clinical trials, which is measuring biological ability. Biological activity can be monitored through in vivo, in vitro, and ex vivo assays, as seen here.
In vitro assaying, for example, on cancer cells, is especially useful because it provides an initial screening of the biological capabilities of a certain compound. Because cancer cells grow rapidly, it is more efficient than using a mouse model, or using, say, a mouse model. And also ethically, it can serve as a first test of the compound's potential cytotoxicity without harming the living organisms or humans. So here at lab, we have numerous cancer cell lines. Most useful to our biological assays are Jurkat cells, a human leukemia cell line, HCT cells, which are a colorectal cancer cell line, and CHO cells, which are a model mammalian hamster cell line. We also have hyperderma cells for mass production of monoclonal antibodies. Okay, let's go into cell viability and its importance in vitro biochemical assays. Cell viability is the number of healthy cells in a sample. Understanding cell viability and proliferation helps to aid the understanding of the mechanism by which certain genes, proteins, and cellular pathways affect cells. Finding cell viability can also help to determine toxicity levels of anti-cancer small molecules. It's because of these means that many cell viability and cytotoxicity assays have been created and are widely used among biologists. Biochemical assays work by measuring structural and metabolic changes or physiological and biochemical activity of certain biological molecules within cells. In order to determine that activity, an assay needs to have a certain readout quality and be rapid and quantitative. Visibility and cytotoxicity assays work by colorimetric, fluorimetric, or bioluminescent detection. For example, the MTT assay, which is depicted here, is colorimetric and can be measured by a color change from a yellow color to a purple one caused by the reduction of MTT to formazan, which is measured by absorbance using a plate reader. Some examples of common optical reporter assays include dyes such as tryphane blue, metabolic activity reagents such as tetrazolium-based assays, and enzymatic activity substrates. One of these optical reporter assays include the MTT assay, which is depicted here, mentioned before, which is one of the most widely used assays for cell viability. The MTT assay is a colorimetric cytotoxicity assay that relies on the reduction of tetrazolium salts by intracellular oxidoreductase enzymes to their formazan counterparts, which, being brightly colored, provide an optical readout for the redox activity in living cells. So basically, biological oxidoreductase enzymes present in the mitochondria take NADH, which in this case is the reducing agent, and reduce it where it is oxidized into NAD+. This assay is called a colorimetric assay because it is quantifiable by the purple dye produced from the reduction reaction. The purple dye is the proxy, or I guess metric, for the amount of living cells. The presence of purple is therefore indicative of an active enzyme indicating an alive cell. As a basic workflow to perform an MTT assay, cancer cells are first prepared and seeded in a 96-well plate with the anti-cancer compound being tested. Then MTT reagent is added, and after periods of incubation and the addition of DMSO, reduction of MTT to formazan crystals occurs, which is then dissolved in DMSO, producing a purple hue. The assay is then quantifiable colorimetrically by measuring absorbance at 500 to 600 nanometers using a multi-well spectrophotometer. However, when performing MTT assays, we observed results that were generally slightly inconsistent. Without any cancer cells present, our media control wells were turning purple, indicating autoreduction. Because of this, we hypothesized that there may be substances within our cell growth media that might be interfering with our MTT results. Here in this picture is an example of one common media component, cysteine, which we then combined with MTT and exposed to base, which you can see turned purple and auto-reduced, producing a purple hue without any cancer cells present. And here, this 96 well plate exemplifies once again the purple coloration change in the MTT assay. So with this in mind, we designed an experiment to test if there was interference in our assay from reductants in the media. Here is a table of reductants found common across many media types, which we screened. We chose cysteine and methionine as model amino acids that could be easily oxidized, which contain a thiol and a sulfide functional group respectively, as well as glutathione, a tripeptide containing a free thiol. We also screened ascorbic acid because it is commonly used as an antioxidant, and glucose, which is a model reducing sugar. We screened glycine as a control to verify that the amine or carboxylic acid of general amino acids are not the specific groups that are reacting with MTT. We then ran MTT assays of these 7 reductants at different pHs and observed results through a plate reader. And then we identified the mechanism of certain thiol-mediated redox reactions of MTT through LCMS. So here are our preliminary results, which we ran in a pH of 8 and a pH of 10. Observably, ascorbic acid, cysteine, and glutathione have higher shown reduction at both a pH of 8 and a pH of 10 compared to the others assayed. We then utilized mass spectrometry or LCMS to determine the mechanism by which this autoreduction occurs, finding that a thiol disulfide redox couple auto-reduces cysteine into cystine. However, the mechanism for ascorbic acid is a bit more complicated and we're currently working on it. In our next trial, we tested cysteine and glutathione have higher reduction, though interestingly, much higher at a pH of 10. What this tells us is that these 3 reductants are likely interfering with the reduction in the MTT assay. To solve this issue in the future, these results indicate that we should either use a media with reduced levels of ascorbic acid, cysteine, and GSH when running the MTT assay, or simply just use a higher concentration of cells to counter the autoreduction. Okay, let's move into some background into monastrol, which was first reported in Science in 2000 from Tim Mitchinson's group from Harvard Medical School. Figure A shows a normal cell in metaphase with chromosomes, which are blue, lining up at the center of the cell. Below that, figure C shows a cell once introduced to monastrol, in which the microtubules invert so that the chromosomes cannot divide properly, which causes mitotic arrest and apoptosis. Therefore, monastrol has been proven to be a potent mitosis inhibitor through its disruption of metaphase. This is depicted again here, where its disruption of metaphase essentially causes the cell cycle to malfunction and the cell to go into apoptosis. And it is because of its inhibitory activity and conduction of apoptosis that monastrol could be useful in anti-cancer small molecule research. Utilizing computer modeling techniques such as DFT and docking, here you can see molecular modeling of how monastrol allosterically inhibits kinesin EG5, which is how it's able to disrupt mitosis. This allosteric binding pocket is flanked by hydrophobic residues, which we hypothesized that with the addition of a trifluoromethyl group at C6 would engage in a hydrophobic binding pocket. So we synthesized monastrol, LASM63, and other analogs through multi-component Viginelli cyclocondensation reactions. For reference, this is an example of semisynthesis, which is basically a modification of monastrol's structure in order to hopefully alter biological effectiveness of the compound in cells. And it's through these multi-component reactions that we could create different analogs by basically mixing and matching components of the reaction to make different analogs. So going back to our original hypothesis, we were particularly interested in fluorination, also because it's popularly used for medicinal studies, mostly because fluorine has a high electronegativity and greater stability of bonding with carbon. Fluorination generally is also known to improve drug metabolic stability and cell membrane permeability. So basically, we made fluorinated analogs such as trifluoromonastrol. You can observe here that we basically added the CF3. Here, trifluoromonastrol is docked, and you can see here that it engages in hydrophobic pocket residues. And here is listed basically the analogs that we made. through gram-scale synthesis and mechanistic insights through 19F nuclear magnetic resonance, or NMR, we were able to make these compounds synthetically in our own lab. This provides us with pretty rapid access to a diverse amount of scaffolds. And so after we made these analogs, I ran an MTT assay on monastrol and two of its analogs, trifluoro and hemi, and then measured it against a positive control of 1% DMSO in cells. However, against expectations, fluorination seems to have made trifluoromonastrol cytotoxicity ability slightly worse than monastrol, which is seen in that the absorbances are higher for trifluoromonastrol than monastrol. So we're hoping to explore this in future studies. Okay, moving into studies of andrographolide, which is a natural product diterpenide extracted from the plant Andrographis paniculata. It is shown to have a significant anti-cancer, anti-diabetic, anti-inflammatory, antibacterial, and more properties. Andrographis paniculata is a terpenoid natural product which gets its medicinal properties from its central alkene, which allows it to serve as a Michael acceptor. Therefore, as a Michael acceptor, it can be utilized in a wide variety of settings, such as in the covalent inhibition of cysteine on a protein target. One important aspect of andrographolide is that it is an inhibitor of NF-kappa B, which is a transcription factor involved in key anti-cancer research pathways. Transcription factors are essentially at the crossroads of cell metabolic activity and can turn related genes on and off accordingly. Transcription factors therefore regulate many important cellular behaviors, in particular, things such as inflammatory responses, cellular growth, and apoptosis. And it does this through alkylation of cis62 at the P50 subunit, reported previously in the literature. Here you can see how andrographolide reacts with the nucleophilic cis62 of the NF-kappa B P50 through a Michael addition computer modeling figure. You can see that in figure B. So once again, this is andrographolide, where you can see the C-ring butanolide, which is basically what drives its biological activity, and the AB ring, which is what enables its binding. We therefore based our analogs accordingly to these key areas. And in order to quantitatively measure the efficacy of Michael acceptor properties of each compound, we monitored the reaction between a free thiol source, which was glutathione, with compounds using a 5,5-dithiobis 2-nitrobenzoic acid, or DTNB, for the colorimetric quantification of free thiols in solution. This ex vivo method is advantageous since the reaction is rapid and stoichiometric. Here you can see the standard curve of glutathione concentration versus DTNB conversion that was used to determine the Michael addition kinetics of andrographolide and our analogs. These results were actually just recently published, and basically, to summarize it, consistent with expectations, andrographolide, which is compound 1 on this graph, and acetonide, which is compound 2, performed equally as well as Michael acceptors. However, compound 5, which was the 11-12 elimination product, was a significantly better Michael acceptor. Compound 3, which was with an acetate in place of a hydroxyl at C14, demonstrated the poorest Michael acceptor kinetics compared to the other analogs. So this is the mechanism of one of our analogs, acetonide, which is a ketol prodrug that was thought to be significant. This is because when you look at the C log P of acetonide compared to andrographolide, acetonide has a higher C log P, indicating that it is more hydrophobic, which we hypothesized would improve its cell permeability. But this is the results of our recent MTT on anti-cancer activity of andrographolide and its analogs, which actually just proves this. Interestingly, acetonide seems to have worse anti-cancer abilities than andro. You can see here that the 11-12, which performed well ex vivo in our previously reported assay, also performed well in vitro. As you can see, absorbance values for 11-12 were lower than andrographolide. But interestingly, acetate, which performed poorest ex vivo, showed the most increase in anti-cancer capabilities in comparison to andrographolide. Another section of this project is our work with RT-qPCR, which we are exploring as a means to find the mode of action by which andrographite derives its anti-cancer abilities from. RT-qPCR, or quantitative real-time polymerase chain reaction, essentially provides us a way to screen gene expression. The basic principle behind this involves reverse transcriptase, which reverse transcribes mRNA into cDNA, which is complementary DNA. The cDNA is then amplified, and the more cDNA you have, the more fluorescence is generated when it's amplified, because fluorescent probes fluoresces with increasing concentration of nucleotide present. In this case, we're using RT-qPCR to determine how genes involved in the NF-kappa B pathway are expressed when andrographite is introduced to the cells. Here is a chart of genes that we chose to focus on, which are all related to NF-kappa B or MAPK pathways, which are related to anti-cancer effects. Determining what genes are expressed more through this assay will basically determine which genes are affected by andrographite, which would help our understanding of the mechanism by which andrographite gains its anti-cancer properties from. Here was one of the first runs that we ran on our DRCAT cells. So because of these results, we can tell that the procedure worked. Additionally, a new pathway correlated to andrographite, the mTOR pathway, was also reported in literature that we're looking into also screening when we redo our RT-qPCR assay. We are currently looking into genes related into that pathway and designing primers so that we could use this for future experiments. And that's all. Thank you so much for listening, and if you have any questions, let me know.
Great job, Kara. That's awesome. Questions? Any questions? So I'll ask the question because I think this is worth mentioning. Kara, how many attempts did it take for you to get a successful MTT assay? And how long did that take?
We've been doing trials of the MTT assay since, I believe, like last December. So probably like a year to a year and a half to finally get it to work.
There you go. So all of you who think that you're so cool for troubleshooting things for 4 weeks or 5 weeks, there you go. 1 year of troubleshooting went into that experiment. Great. Questions, comments?
Well done, Kara. 50 slides. Aim it to 20 for next time. I know it is hard. I know it is challenging. But sometimes when you grow in the research community or you want to present something, you just have to be very precise, like very few slides and deliver what you want to do. So just practice. I know it is hard. Trust me. I'm very talkative and I create a lot of content. I totally feel you. But the fact is bitter. So I know it's not fun, but next time, give it a try. Well done. Good job.
Okay. Great job, Kara. I think we are back to Sahar's group. Is that right?
I guess so. I can see the team is here. Let's see what they have. Okay.
All right, I'll get to screen sharing.
So welcome back to our presentation, guys. I hope you're all enjoying Colloquia tonight. We have some really exciting data to share with you. So now I'm going to go into the actual details of our research study. Essentially, our general research question is, how does the COVID-19 pandemic affect the behavioral plasticity of adolescents? The purpose of our study is to collect information on the dissonance adolescents have experienced over the course of the pandemic, and to understand the correlation between the effect of the pandemic and dissonant feelings teenagers have recently experienced. We hypothesize that new and constantly changing legislation concerning vaccines, masks, public safety, and school regulations have led to students experiencing cognitive dissonance. Furthermore, we hope to get around 200 participants enrolled in our study. So our process of data collection consists of two parts. The first part is consent. Basically, we first send a long consent form to students' parents. Once the parents fill it out, a short consent form is sent to the students. And once the students fill it out, they can then complete and submit the questionnaire. Now, I know I touched on this previously, but the questionnaire essentially asks adolescents about their opinions on COVID-19 vaccines, regulations, school restrictions, and masks. Participant identity is completely anonymous. As soon as participants submit the questionnaire, the response is linked to a randomly generated ID, which we will then use for data analysis. And so here is a short timeline of our research study. So we first found and read numerous papers and previous literature on COVID-19 and cognitive dissonance. Then after extensive research, we created questions for a questionnaire and drafted it on jot form. Then in order to be able to extract quantifiable data for our survey, we then created our own scoring protocol for measuring participants' dissonance. Then we drafted and sent our study proposal to IRB, the Institutional Review Board, as well as drafted the long consent form for parents and short consent form for students. Currently, we're in the process of data collection for our study. We've sent the long consent form out to ASDRP parents and the parents of friends, and are currently trying to encourage as many adolescents to participate in our study as we can.
All right, so now I'm going to get into the preliminary data that we have collected as part of our study and talk about our analysis of the data that we collected tonight. So here are some graphs of the demographic data that we collected in our preliminary round, where we invited members of the Colloquia Neuro Lab and our family and friends to participate in our study. We had 32 consenting participants and 49 parents who consented as well, 13 of which were male and 18 of which were female. One individual preferred not to provide their gender. 97% of our responses came from Asians and 3% were from people of Middle Eastern descent. 78% of participants were fully vaccinated and 90% had no booster, while only 3% had only one shot. The average age of a participant in our study was about 15.857, so almost 16 years old, which is what we were aiming for as our study is targeted towards adolescents. So if we take a look at the dissonance scores that were calculated from our data, we can see some trends begin to emerge. For perspective, a dissonance score of lower than 4.5 for one group means that the dissonance was significant. We can see that significant amounts of dissonance were calculated in the general COVID field, the government and authority field, and the schools field. According to our calculations, people faced the lowest dissonance in the vaccines group, and they felt the highest dissonance in the government and authority group. If we put this data into the lens of the real world, we can see that people in our study had less problems acting consistently with how they felt about vaccines than they had about acting consistently with how they felt about government interventions dealing with COVID-19. There are still many other analyses that we plan to run on to see how people's reactions to one group correspond to their reactions to others. These analyses may reveal patterns of how people process information regarding COVID. For example, people who experience less dissonance when it comes to vaccines could be more likely to experience less dissonance regarding masks as well. However, at this moment, we have not completed this part of our data analysis. So here's a summary of the data that we collected across all of our categories. As you can see here, participant 13 had the lowest dissonance score, and participant number 6 had the highest dissonance score. Overall, almost 72% of people faced a significant level of dissonance overall, and 75% faced dissonance in group 1, while 40% faced dissonance in group 2, 53% faced dissonance in group 3, and 78% faced dissonance in group 4, while 75% faced dissonance in group 5. Again, there's still much more analysis to be done on the data that we've collected to find an understanding that can be applied to the real world. So now we'll get into some of the new data that we collected today. So overall now we have 46 responses to our questionnaire, including 14 new responses from you all today. With these new responses factored into our data, we have 60% of our responses coming from females, while 38% come from males and 2% come from people who prefer not to provide a sex. In terms of race, 93% of our responses have come from Asians, while 2% have come from people of Middle Eastern descent, white people, and people of races that we did not list in our questionnaire. 81% of responses also came from people who were fully vaccinated, while 17% came from those without a booster shot, and 2% came from those who only got one vaccine shot. Finally, the median age of our participants was 15.7 as well, which was very close to what we saw in our first group too. So in our new data that we collected tonight only, we can see similar trends to what we saw before. For the general COVID-19 group, the average score was about 4.46, for vaccines, 5.14, for masks, 4.98, for the government and authority group, it was 3.26, and for school, 4.06. So as you can see, the vaccine group still had the lowest average distance score, while the government and authority group had the highest. Although with so few responses, it's not possible to make wide generalizations about society, this data still points to people having fewer problems acting consistently with how they felt about vaccines than they did about acting consistently with their government interventions dealing with COVID-19. So as we can see in this graph, among tonight's participants, 57.1 experienced significant distance again, and participant 33 had the lowest overall distance score, while participant 36 had the highest overall distance score. Overall, tonight's participants had a lower distance score than responses we collected before, where almost 72% of participants in our original group faced significant distance, while only 57% faced distance tonight. And that basically wraps up the data that we collected today, so that brings us to the end of our presentation. Special thank you to Mr. Harja, Hanakia, the whole distance team for getting all this data collected for us and analyzing this so quickly tonight, the ASDRP admin and the All Children Foundation for helping us conduct this data as well. We're happy to answer any questions you might have. Thank you.
Thank you. And that will bring one more very important question, that why research is so slow and why we're still behind answering so many important things in the world, seeing that there are 234 participants here. And out of 234, there's just 14 participants presented. So even though we are participating in research, even though we are doing active research, even though we talk and breathe research, research is still needs a lot of education behind that. People got to at some point trust research, because everything that we talk these days are data driven, and if we don't have enough data. So what I understand from this study, that we need to go to high school, and talk that we need to educate kids and talk with them regarding vaccines. We've still not been clear what this vaccine means. That's why they're facing a lot of dissonance. That's something that the counselor needs to spend time on it. The parents, the education, the investment of the society should be on something like that. So I think that's a big lesson for you guys, that even though there are over 230 people here, getting us 14 data points is very valuable. We appreciate those. We still have a long way to go, but just a quick reminder that where we are, how we are looking at the society. Again, unfortunately, research is still way too behind at what we are thinking. We would have got so many drugs done. We would have got so many solutions to so many problems done if research would have found its way to explore more, and people would have trust research. So research is not scary. We are not misusing data. We are trying to answer some questions that what needs to be answered, and that is a real life problems. For next 10 years, we have to yet understand much more about COVID-19. Again, we study, we continue to hit our target. We have a long way to go. Thank you so much for all of you to listening to my awesome team. There is a lot of hard working behind it. A lot, and trust me, getting those data analyzed and getting for you, these guys are just amazing. They are back. We have thank you so much for people who help us, video trap and they're backstage. So they're a backstage team working. There's a front people, front end and back end. So thank you all of you. It just giving you a real life experience of what is research, how it is done, and what it takes to do human study and just give you a sense of real life. I was anticipating, I was telling them 10, I think 10 people will participate, but I wanted that you see what is a real life research and when we say time consuming, when we say research takes time, participation takes time, analysis, as previous team mentioned, that needs a year. It is not easy. So welcome to the real world. Thank you so much for listening to my team. Thank you to my awesome team to give you this real life experience. And we don't have anything else. We'll see you next time.
Great presentations. Let's see, Ananya, make sure your parents check their spam. Sometimes our ASDRP emails go to people's spam or into their promotions tab. So have them double check.
Have a good night.